Northern blotting, also known as northern hybridization, is a molecular and genetics technique used for the detection and quantification of specific RNA levels using a hybridization RNA probe. Northern blotting involves three major steps. Separating the RNA, cells or tissues are lysed and RNA is extracted. Isolation could be done using the acid guanidinium thiocyanate phenylchloroform extraction method. RNA is then separated by agarose gel electrophoresis. The separation is done based on size. The formaldehyde present in both the gel and in the migration buffer ensures the inhibition of ribonuclease activity. The RNA is now negatively charged. Transferring the RNA. The RNA is then transferred to a nylon membrane. The membrane, driven by capillary forces, is exposed to intense ultraviolet, UV, light to induce RNA cross-linking. The RNA is immobilized. Hybridize the probe to the membrane. The probe, which is radiolabeled, is complementary to the gene of interest. The membrane, affixed with RNA, is hybridized with a radiolabeled probe, forming a double-stranded RNA-DNA or RNA-RNA structure. The membrane is washed to get rid of nonspecifically bound probes. The membrane with the probe specifically bound to the RNA is then dried, exposed, detected, and analyzed by autoradiography or using a phosphorimager apparatus.